Hey guys, and welcome back. Welcome back. Welcome back. So, today we're going to be talking about facilitated diffusion and really the segue to active diffusion. So, 5.3 is talking about passive transport, and now we're looking at some active transport. So, active transport, as you would guess, is going to require energy. And so, that energy that's needed to do this is going to be provided in the form of ATP. ATP is typically called adenosine triphosphate, and it's abbreviated to ATP, and that's something you should know the two. ATP is adenosine triphosphate, so there's three different phosphate groups that are going to be chained together. That last phosphate group is going to be broken off again and again. Whenever it is broken off, it is no longer a triphosphate. It is two, as in diphosphate. So it is ADP instead of ATP. So as we're looking at active transport, there's certain molecules that are going to be involved, certain proteins that are going to be involved in going against the concentration gradient. So the concentration gradient is going to be something that's going to be identified as where we have a higher concentration to a lower concentration. Now, that passive transport going from higher to lower is quite natural. Going the other direction is going to be called active transport because this is when things are going to go against their concentration gradient and they're going to take energy to do that. So, if you think of being in a stream, better yet, a river, you can have a life preserver on and you could even doze off and you'll travel downstream. That's more of a passive transport. No energy is needed to do that. For active transport is going upriver. And so that's you going against the grain, you going against the concentration gradient and moving towards where there's higher concentration. That is not a natural process and it requires energy to do that. A classic example of a protein that does this is the sodium potassium pump. Sodium potassium pump is, I would say, the most known type of active transport that we're looking at protein pumps. And so let's go ahead and look at a little bit of how it works. So here we have a concentration, and you can do just an overview and see that there's more sodium on this side than this side. And so there's a natural tendency for sodium to move in this direction. You can also look at the amount of potassium decays, and it's going to go from a higher concentration to a lower concentration unless you're going to use something and use energy to do it. So this is a sodium potassium pump. Notice the roundish areas that the sodium will dock into. And also appreciate here that the potassium is going to be these little triangles. They don't fit. Now, you notice there that ATP was used, and you have the inorganic phosphate that's there. When that opens up on this side, the potassium quickly fills those locations. You can see sodium that's going to load. You're going to have ATP that's going to be used, and you're going to have this again opened up. As a result of this, this changes the shape of these, causing these to be ejected. And now the sodium can go out, and the potassium can actually load into these pieces. The loading of the potassium is going to allow for it to go against the concentration gradient. Once it has that conformational change here, it's going to cause these to be decreased in size, causing them to be ejected on the inside. Here is a structure of an ATP synthase. In this ATP synthase, you can appreciate a few of the things here. This is showing just more of a molecular view. You can see the alpha and beta helices there, and you can also see the alpha helice here. We have ATP that's going to be moving in to be bonded. As a result of that, you're going to have a conformational shape change, and you're going to allow some of the work to actually take place. This video is a really good video, and I'm going to provide this through a YouTube link so you can watch it. It's about 4.5 minutes, but it's really good, and it shows all of these pieces kind of working together as you look through the actual structure of the phospholipid bilayer and the way these different protein channels work. So we looked at this just a moment ago, but let's go ahead and pick a spot. And since it's cyclic, it doesn't matter where we start. We have the sodium that's docking into this area. And notice that the potassium is really small. You can't bond there. As a result of when these bond in, you have ATP. It becomes ADP. You have the inorganic phosphate that's going to be there. As a result of this bonding, it's going to cause a shape change that's going to cause it to hinge, which is going to release that sodium to the outside, collapsing the structure of the sodium, which causes them to release And you also have the opening up of the potassium, which then allows for a place for them to bond. As a result, they can bond, and when they bond, that is going to cause it to hinge back, and you can release the potassium back to the inside. But when we're looking at passive transport, we can do just diffusing through a membrane. That's just diffusion. And osmosis is going to fall here as well, going from high water to low water through a selectively permeable membrane. Here we have facilitated diffusion. You can have higher concentration to lower concentration. This is a channel protein. An example would be an aquaporin. And here we have a carrier protein that's going to hinge back and forth, going from higher concentration to lower concentration. This looks a lot like that. But the way it works is that this goes from a low concentration to the higher concentration. It requires energy. That's why it's called active transport. So, membrane potential. Membrane potential is going to be the potential of the voltage that cross the membrane. And why you have this membrane potential is, if you think of it, you have ions that are on one side, a higher concentration of ions one side to the other. As a result of those charged particles being on a side, it's going to have its own literally electrical charge. The voltage that you have between the two sides are going to really be an important part of driving a lot of these electrochemical concepts. So, electrochemical. First, let's kind of identify the two parts of it. And one is the chemical force, and that is the ion's concentration gradient. But you also have an electrical component. That electric component, as you would guess, is going to be due to charge. So it's able to move from one side to the other based on charge. So some ion channels are going to be gated ion channels that only open under certain electrical charges. So this electrogenic pump is going to be a voltage, generates a voltage across the membrane. So let's look at a proton pump. A proton pump, as you would guess, is going to pump those hydrogen ions from one side to the other. And it's going to do that as a way to create potential energy. And so it's going to spend some of this energy, this ATP, to create a gradient, to create a larger concentration on one side to the other. And that's going to be used as a co-transporter. So here we have that protein that's going to be pumping the hydrogen ions across. And you're going to see now a large amount of hydrogen ions that's here. And you also notice that's going from lower to the higher side. And so it's going to require energy to naturally do that. Now notice we also have the hydrogen ions. We have a charge here, plus. By default, this is negative. So because of this charge difference, we're going to have a few things that are going to take place in this co-transport method. So the co-transport, this is quite common, and we have this happening a lot in cells. And this is where you have hydrogen ions that are going to be used by this proton pump to generate a large concentration that then are going to naturally, passively move back in. So here we have that. We have that proton pump that's pumping them out, creating this proton positive electrochemical gradient. And you have something like sucrose. Sucrose is going to bond to this sucrose proton co-transporter, where once it bonds in, these are going to move naturally in this direction. And so it's going to be a co-transporter in the sense that when this is docked and this goes this direction, think of a turnstile they have at malls and different places to count people. Once that turnstile is going to be pushed forward, other things are going to be moving with it. And so this is occurring here. So think of it as something is going and bonding here. And then as a result of that, it's going to naturally spin. And when it spins, it's going to release the hydrogen ion on this side, going from a higher concentration to lower concentration. So this is the diffusion of hydrogen ions. And it is utilizing the sucrose to get it in. And so this doesn't have a thought in the process. This is just perfectly designed in the sense of evolutionarily significant, that this docks here perfectly. And as a result of docking there, and this hydrogen ion that's going to then cause this to spin, it's going to cause sucrose to be on this side of the cell. So by generating a proton gradient, you now have the diffusion of these across. You're going to have the sucrose, a hydrogen ion co-transporter that's going to move sucrose in passively. And it's going to do that through the process. And it's even showing a lower concentration to the higher concentration. It's going to do that in the process due to the diffusion of these hydrogen ions across that membrane. All right, guys, so that covers 5.4 with active transport. We'll get into more active transport soon. Thank you.